2 and its detection procedure. The first procedure I have already discussed with you is all about RT-PCR method, which is an extensive method giving us a total data not only about the person's tested positive but also a detailed analysis of his or her how much infection he is carrying from the CT value. Now in the second method, it's a blood test method. In this method, first only I want to say it's a rapid test method. It's going to be happening within 10 to 15 minutes. You can test from the kits that are available in the market. Normally a needle prick from anywhere, you can take it from the finger, a needle prick, a little bit amount of blood, and then there is a kit which resembles somewhat a pregnancy kit type of thing. You can see I have already drawn the kit for you. This is the region where the buffer solution is given. The kit is supplied with a buffer, and the sample, that is the blood sample, is going to be put over here, and there are specific areas. There is a control region C. You can represent it with C. Here it is IgM and IgG. These are standing for the primary and the secondary immune response antibodies. So if it is primary immune response antibody, then it is standing at IgM, and if it is secondary immune response antibody, then it is standing at IgG. So coronavirus has a specific antibody. This virus has a specification as far as antibody is concerned, but it is not able to detect whether it is SARS-CoV-1 or SARS-CoV-2 because these are familiar, it reads as a familiar family virus. As I have said, it resembles the pregnancy test kit, and there is a control line you can see. When you run the test, means you put a little bit of blood sample over here in this region, which stands for the region of sample, and then you put a little bit buffer. You have to put the buffer. The kit is supplied with a buffer solution. What happens is, due to capillary action, blood has also got its antibodies. Due to the capillary action of the buffer, and it carries the entire blood components, and it reaches the point where the control line is there. So if there is a control line, that means it is signifying the fact that your kit is correct and the test is already run. Next fact is, virus means each and every virus has a specific antibody. It means it is able to recognize, it is signified a particular antibody. In case of coronavirus also, there is a specific antibody. Antibodies generally we denote it with the symbol of Y kind of thing. And what happens is, if our body, just let me give you a hint, what really happens is our body has immune system. This protects us from diseases. Now what happens when this virus attacks our body, means it takes us as a host, our body, which has produced antibodies, is going to attack and kill that virus. So SARS-CoV-2 has a particular antibody. So if that antibody is present in our body, that means at some point of time we had already been infected by SARS-CoV-2. This virus has a particular antibody, but it is unable to detect which SARS-CoV. Is it SARS-CoV-2 SARS-CoV-2 or SARS-CoV-2 or SARS-CoV-1? That it is unable to detect because it recognizes as a similar family virus. But if that antibody is present in our body, it is enough a proof that yes, you had been infected by coronavirus. So let's see how the kit, I'm going back to the explanation of the kit. As I have said, we put a little bit blood over here, little prick, we put it over here, and then we add the buffer solution in it, and we run the test. And we find the line, we have to wait for 10 to 15 minutes. Normally plasma, which is the watery content of the blood, is to be taken. But at home, if you do this experimentation or testing, it is not possible for you to extract the plasma and do it. So we are going to do with the whole of the blood. Due to the capillary action, this buffer is going to carry blood and its components across the kit. So if you find a line at the control section, it is a proof that the test is run and it has means that there is no problem as far as the kit is concerned. Now if you find that your test has run, it's completed, but there is no line in IgM or IgG. IG stands for immunoglobulin. And this signifies the primary immune response antibody. So if there is no line in the primary immune response antibody or the secondary immune response antibody also, there is no line, but at the control section there is line. That means the test is run, you are out of danger, you are not tested positive, you are negative, you do not carry SARS-CoV-2. So the test is finished over there. Now if you find that yes, your test is run and you find there is a line in IgM, primary immune response antibody. That means in the very recent time, almost 1 or 2 weeks, or currently also, you are carrying the infection, SARS-CoV-2. So immediately you must go to the clinic and take the necessary steps, that is self-quarantine, isolation, because you are the person who is going to carry it to others. So you can transmit. Next we come to, as I am finished with C line and IgM. Now I am talking about the secondary immune response. Suppose you run the test and you find there is a line in IgG, that is secondary immune response antibody. That does mean that a couple of times you have been infected with this virus earlier, much earlier. This infection was there and now you have developed some immunity and you are in a safer state, but you are still positive. So this is a concern, IgM and IgG. But if you are running the test along, means you find a line in IgM, IgG and in the control section, which is showing that the kit is proper. So if you find a line in IgG and IgM, that does mean this virus has attacked you earlier because it is secondary immune response antibody. So you have been kind of immune also. You have, if without mutation that virus attacks you again, you are kind of immune. But at the same time, your primary immune response antibody is showing the result that currently you are carrying this virus. So you are in a good position to transmit the disease. Naturally, you should be very careful and all the initiatives must be taken. So, as I have explained, this blood test method is a very rapid method, 10 to 15 minutes it takes to run it successfully. You can even do it at home. It resembles the pregnancy test kits only. But when you are performing this, keep it in mind, neither the RT-PCR test, which I have explained in my other video, nor this one, the blood test, is going to give a standalone result. So results might be faulty. Maybe there was some problem with the kit. In this case, maybe the buffer solution was not a problem, it was not working properly. In RT-PCR, I said that there are several factors, even reagents are factors. So properly, if anything doesn't work, slightest mistake also can cause negative results, false negative, false positive results. Naturally, a standalone method, in my earlier method also, RT-PCR have specified, unless and until skilled technicians, proper lab means collection of samples, storage, transportation, reagent contamination can take place, buffer may not work. So several problems can be there. Due to slightest mistake, a false result can be there. So IgG, IgM, these are antibody tests. Coronavirus has a specific antibody, but it is unable to detect which antibody means which is the virus is it. Is it SARS-CoV-1 or 2? Because it resembles, it reads as, it recognizes as a similar family virus. At the same time, one more point I would like to keep in mind that whenever SARS-CoV-2 virus is attacked, there is nothing to panic about it. Let us all join hands to fight this global crisis. Do practice the social distancing and stay tuned with my channel, bringing on more videos. on the board. It is Sputnik V vaccine. The idea of this vaccine, before I speak about it, let me tell you one thing, that this is not the vaccine that is soon launched for the very first time. In India, of course, it is coming for the first time, but earlier it was in last year, 2020. Already in August, there was the vaccine had been launched in Russia. So this is the vaccine which has already been launched in Russia, and right now in this month, current month, that is April, we are thinking that India has been on this situation that we are right now thinking about this vaccine, Sputnik V. Before I talk about the mechanism of this vaccine, let me tell you one thing. What is the idea of vaccination? See, in our body, when any pathogen, that is any germ, is attacking our body already has a proper immune system, and this immune system is protecting our body by producing antibodies. Let me draw the antibody diagram like what we denoted, that this Y symbol is denoting antibodies. Antibodies are also proteins which are known as immunoglobulin. Our body's immune system has two parts, you can say. One is the innate immune system, which is not a very specific one, as it is acting on all the germs in a similar fashion. So initially any germ, which is on the skin or maybe in the tissues or in the bloodstream, it is fought by these you can say right now we are not talking about the secondary immune response, so I am not talking about antibodies, I am talking about the initial fight, that is by these cells which are fighting, and these are going to fight against the germs. That is the innate immune system, which is non-specific. It acts in a general fashion. But if I am going to talk about the adaptive immunity, rather we can call it adaptive immunity or acquired immunity. So acquired immunity or adaptive immunity, you can say this is the part of secondary response. It is the part of, I write in this way, so I am not talking about the innate. The innate is the first response that is given. Okay, generalized manner, non-specific it is. But in case of the secondary immune response, it is adaptive immunity or you can say acquired immunity. These here the cells are very specialized and specific in their function, and these are conducted by WBCs and T lymphocytes and the B lymphocytes. So in my discussion, I am not going to focus too much on this topic. I am going to focus on the vaccination. Vaccines, generally what I am talking about, the antibodies. Antibodies are parts of acquired immunity or adaptive. Let's focus on adaptive immunity. Here the cells are very specific in their function, and antibodies, which are also proteins, and these are acting specifically. So in this case, what I am talking about, adaptive immunity or acquired immunity, is conducted by the antibodies, that is WBC, the T lymphocytes, and the B lymphocytes, B cells and T cells we call it. And here this function is very specific. What happens is, after an initial attack, when our body is being attacked with any germ, the pathogen, any foreign particle, a virus which is always present, it may be present around us, but our body has got a defense mechanism. The first line of defense is given by this innate immunity, which is not very specific, non-specific it is. But in case of secondary immune response, it's very specific. It has a good immunological memory. That means it remembers its enemy. It has a good immunological memory, and it remembers its enemy very well. That yes, this is the enemy I had fought earlier, and then it binds with the
antibodies have a big responsibility. It binds with the pathogens. In this case, the pathogens which are around found, that is antigens, it binds with it so that the host is safe. This is the whole idea, and this idea is actually the idea of vaccination. Vaccination is artificial inoculation. In this case, what we are talking about, when any pathogen is attacking our body, it is the body's secondary immune response, the antibodies, which are proteins, immunoglobulins. These are binding on to the antigens and they are fighting. Henceforth, the virus or bacteria is going to be killed. This is the total idea, and this idea is by vaccines. Now you may ask me how it is going to happen. Many people have a false idea that why we are going to take, we will not take vaccines. Why? Because we are going to, our body is going to be fed with virus. Yes, it is. Draw the diagram in this manner that any virus, it has got a structure. You may have seen in diagrams right now in many places. We are very familiar with coronavirus diagram. So it's something like this, and it has got the genetic material inside it, which is RNA. In case of coronavirus, the genetic material is RNA. It's RNA virus, and it has got some spikes. Let me draw the spike with different color, so that it is easily understood. It has got spikes like this, the spike proteins we say, and there are several receptors. These are the surface receptors. Okay, so it's on the virus will be identified on the basis of some of the characteristics, and one such characteristic is the spike protein. Okay. Now the structure is something like this. Coronavirus, which is where the genetic material, a virus can only harm you if it is able to replicate, that is reproduce, and it only replicates when it is inside a host which is living. So if a living host is present and it somehow enters the living host, then only starts its activity. And in this case, coronavirus, the genetic material is RNA. So when we are talking about vaccines, if I am going to introduce the virus into a person, the person will be showing the disease symptoms and will be attacked by this virus. So this thing I cannot allow to happen. So what our scientists have thought, that why don't we do something, a mimic of the whole incident, that we are introducing a virus which is harmless. So before you have this thought that I am going to take a virus and so I am going to have something which is harmful to our body, no, not at all. Your body will be completely safe after vaccination, provided that you are following certain protocols of it. That is, in this case, the virus which is carrying virulent properties, that is the factors that can cause disease, and that is from the proteins. Virus will be will be producing the transcription and the translation, and finally proteins. These proteins are responsible to carry the diseases or the disease causing factors. So if we remove those disease causing factors and then we weaken the virus, we have to weaken this virus in such a way that it is harmless. Remember, I am putting it in this box so that everybody can understand this fact, that the virus that is given to your body is completely harmless. This is a vector that is used, is adenovirus, where the small component in red you will find this is the SARS-CoV-2 component. Now when this vector, which is a recombinant you can say, is introduced into human body. Now this will be introduced in the human body. This is the human body. This is the adenovirus, which is DNA virus, and this is this component SARS-CoV-2 component, which is RNA virus component. Not the whole of the virus is introduced, just a small component which is able to produce the spike protein, and the virulent factors will be removed. That means that the factors that will produce COVID-19 will be removed from it. The virus is now weakened. So this will be introduced into human body. This is the human body. What happens is our human body, the adaptive immunity, which has got the role of it remembers its enemy also. So what it does is it produces antibodies. Okay, the human body will be producing antibodies, which are also proteins. These antibodies, let me represent it like Y. It will be attacking this virus, the spike proteins, and what is happening is when it will be the spike proteins are these are proteins. These will be binding onto this ocean, and these antibodies are binding onto the proteins and killing the SARS-CoV-2. So this is the whole idea of vaccination, that instead of killing the host is human. Instead of killing the host, what is the idea? That when this is our vaccine, when it is introduced onto the human body's secondary immune response, it is the responsibility here. The antibodies will be attacking, it will be binding onto the proteins, the spike proteins. It will be the identification of the spike proteins is in such a way that antibodies will be recognizing it. Yes, this is the SARS-CoV-2, and it will be binding onto the proteins so that our host is free. That means human being is free. This is what is happening in the mechanism of Sputnik V vaccines. Now what happens is, in a problem about a vector-based vaccine is that in due course of time, means when the antibodies are attacking the virus, it happens that it starts attacking the vector also in the second dosage. Remember in Sputnik V, there are 2 doses. So in the second dose, what the problem will exist is that it starts attacking the vector itself. Now that we have known that our body's immune system is able to recognize the spike proteins, the spike proteins will be recognized as, remember, the spike proteins will be remembered as what? As this is going to be the SARS-CoV-2. So it will be recognized by the human body's immune system, the antibodies. It will bind to the spike protein, and henceforth it kills the virus. Now in due course of time, as I have discussed the problem earlier also, the problem is happening when there is a second dose. First dose, there are 2 doses. Remember, in this case there are 2 doses. First dosage, these antibodies are ready and it will be killing the virus. Now in the second dosage, what happens is, in a vector-based vaccine, this is a vector-based vaccine. Covishield is also a vector-based vaccine. A vector-based vaccine has the problem that in the second dosage, when the antibodies are already ready, means that the antibodies are already ready, it again, when we introduce in the second dose, it identifies the virus SARS-CoV-2. Yes, this is the SARS-CoV-2, and it starts attacking. And when it starts attacking, it attacks the vector itself. That means our idea will be failing in the second dose. So what the Sputnik V, the scientists, the researchers, what they have thought is, they have introduced 2 different variants of this virus. One is RAD26S, another is RAD5S. So these are the 2 variants of the virus that is used, so that in the first dosage, when it is able to identify, in the second dosage, a separate variant of the virus is used. That time it is unable to identify it as the same one. So it will only kill the SARS-CoV-2.
So in the first dosage and the second dosage, you can say a gap of 21 days is maintained. Earlier it was believed that if there is a long gap, it will be better, but now the thought is different in this matter. The Russians have, in Sputnik V, the mechanism is being conducted by 2 different variants. So in the first dosage, the antibodies which are attacking the SARS-CoV-2, which is very specific in its idea, is a secondary adaptive
adaptive or acquired immune response, you can say. So when it is attacking and it is binding onto the spike protein, killing the SARS-CoV-2 component in the second dosage, so that our vector itself means it will not only kill the SARS-CoV-2 component, it starts killing because it has recognized it. It will also recognize the variant that yes, this was the variant I had killed earlier. So that it is not killing our vector, the different variant is used, that is recombinant adenovirus 26S is one variant and the other is recombinant adenovirus 5S. Another variant is used so that our vector is safe but the SARS-CoV-2 component is being killed, the spike proteins it binds to and it kills the virus. So instead of waiting that which vaccine is best in the market, go and take the vaccine. This is the Sputnik V vaccine. We are hoping for the best thing to happen. So when it is going to come, there is a probability of having this vaccine in the last 3 to 4 months of the year. So before that, the Covishield has already come in Indian market, you can say, and Covaxin is also there from our Bharat Biotech, and Covishield from Serum Institute of Pune. So instead of waiting for the best vaccine, please go and take a vaccine and keep yourself safe. And this is the whole idea of the Sputnik V. Another exclusive idea of Sputnik V is also in this matter, that this vaccine is a freezed vaccine. That means it is in powder form. When it will be given to a person, it will be hydrated. This is another idea, and there are almost 4 pharmaceutical companies which are in partnership so that we can have more number of doses, means more number of doses prepared to give us. So in this case, you find Sputnik V. This is all about Sputnik V at this moment. If you want to really know about Covishield and Covaxin, please go and watch my videos and give you likes, and if you want to write anything regarding this matter, please write to me. Feel free to write, give likes, and do subscribe to my channel. Thank you for being with me. Another video tutorial class, you can say, and today's focus will be on the 2 vaccines that have already been introduced in India. One is the Covishield, the other is Covaxin. Earlier I have also prepared a video on Sputnik V, this vaccine. Here I am going to mainly focus on Covishield and Covaxin. So let me first talk about the vaccine. Covishield is Oxford AstraZeneca, a vector-based model of vaccine, you can say, and this is manufactured by Serum Institute of Pune. So in this case, the vaccine is a vector-based vaccine which is based on Oxford AstraZeneca model. The model is quite simplified version of adenovirus. This is adenovirus, a DNA virus. Adenovirus is a DNA virus isolated from chimpanzee, C68 from the lymph nodes, and this virus is a DNA virus, and it's a common flu virus. So after vaccination, the normal symptoms that you can come through or go through will be that of the common flu virus. So there is nothing to feel afraid about it. Mild headache, a little bit of fever for 2 to 3 days. Even without no medicine is required to cure this. So this virus, whenever a virus attacks our body, a virus is generally known by some of the characteristics. It cannot cause any harm to your body unless and until it is able to replicate inside your body. So when it is introducing its genetic material to the body, then only it is able to replicate. So what we do in this is, the virus in open air is absolutely harmless. As soon as it enters our body, a host, a living host, it starts its replication. So what we are doing is, in case of vaccination, the idea is to develop adaptive immunity, a secondary immune response, so that it lasts for a longer time, and when the real virus comes, it is able to produce antibodies to kill the virus. This is the whole idea of vaccination. So what AstraZeneca has done, the Oxford model is all about. This is the adenovirus, which is a vector that will be carrying. It's only the surface. You can say the surface here. You can see the model is something like this adenovirus, which has got some surface receptors, the spike proteins. The alteration is only over here, that only the coat protein, the protein coat that is new. This is the protein coat which has been modified, and here the modification stands in this matter, that it is the protein code, which is that of the SARS-CoV-2. So only a protein code or the spike protein will not be able to, only the protein code when it is introduced, that of the SARS-CoV-2 will not be able to cause any harm to your body. It's just this sport protein that will carry the SARS-CoV-2. So it's not that the whole of the SARS-CoV-2 will be given to your body. It's only the surface coat or the surface receptors that of the SARS-CoV-2. So the genetic engineering lies in this fact. This is adenovirus, which is acting like a vector. It will be having the surface receptors that of the spike proteins, that is of SARS-CoV-2. So this is the only alteration that will be used, and this is our vaccine which will be introduced onto the human body. Now what happens is, when it is introduced into the human body, say this is the vaccine which will be introduced into the human body, and as soon as it enters the human body, it should start its activity. So what happens, our body immune system, this is the body which has got its immune system, the antibodies are going to bind to these spike proteins so that our host, that is the human body, is kept safe and it kills. So this is the whole idea. And in this case, the SARS-CoV-2 only, what it will be identifying? Our antibodies will be identifying it as SARS-CoV-2, and it will bind to the surface receptors, the spike proteins, and it will kill the virus, so that the antibodies can keep the whole of the body safe, because this is the enemy for the whole body which is entering the body, so that in future, when the real SARS-CoV-2, when the real coronavirus enters, if it enters, it will be able to kill. The whole idea of vaccination is that our body's immune system is already provided with these antibodies, which have got a good immunological memory also. It will keep in mind that yes, earlier it has happened that this virus I have attacked and killed, and the second time when the same virus, this time when it will be the real virus attacking, it will be able to kill the virus. This is the whole idea of this vaccine. See, the efficacy rate of this vaccine as given by, as the information has been provided. Normally it is 70.4 to 70.6%. You can say that it is 70 to 80%. And as this is adenovirus, the origination of adenovirus, that is chimpanzee C68, from the lymph nodes it's taken, it can maximum, what can happen to you? A common flu symptom. It's a common flu virus. So do not think that you are going to be harmed by any means. It is refrigerated and kept, and this vaccine has 2 doses. You can say 4 to 12 weeks, or you can right now find out that 6 to 8 weeks gap is maintained, so that the body, from the first dose to the second dose, so that in the first dose, when the antibodies are prepared, in the second dose, a longer gap is maintained, keeping in mind that means this is a generalized view that the antibodies which are already prepared by our body do not cause harm to the vector itself, because in due course of time it might happen if the gap is not too much, then it may attack the vector itself. It is thought, because it has identified its enemy very well. But right now the thought is a bit different, you can say. So the whole idea of Covishield of AstraZeneca Oxford based model is on the basis of adenovirus, which is having only the surface coat protein, that of SARS-CoV-2, which is nothing to feel afraid and with minimum side effects. Is Covaxin. Earlier I have talked about Covishield and now Covaxin. Covaxin, which is manufactured by Bharat Biotech Hyderabad and ICMR, that is Indian Council of Medical Research. Now this vaccine, that is Covaxin, here the idea of this vaccine is a bit different. In case of Covishield, it's all about the adenovirus, where the surface protein, the coat protein, is that of the SARS-CoV-2. In case of Sputnik V, there was genetic engineering. Both Covishield and Sputnik V are vector-based vaccine. In case of Sputnik V, there it was the adenovirus, where the genetic component was altered slightly, and only the SARS-CoV-2, a small component was used and this vector was introduced into the body. But this vaccine is different. Co-vaccine is different because here is the same SARS-CoV-2 virus that is used. Now you may think if this virus is going to be used in vaccine, then we are going to be under attack of COVID-19. No, it is not going to happen because here the SARS-CoV-2 virus that is used is weakened. That means it is made weak or disarmed, you can say. How that is going to happen? Either you heat kill it, you kill the virus heat killed, or you treat with some chemicals or radiation. So what are the ways by which you can weaken a virus? Weakening or disarming the virus of SARS-CoV-2 so that we do not have the virulent properties, the properties that can, like a virus is only able to replicate when its genetic material is inserted into the body. In this case, we are going to introduce the same virus of SARS-CoV-2 by killing it either by heat or with some chemicals or some radiation that will be used to destroy the virus, and then it will be introduced into the body. And this vaccine has efficiency level, it is said, that of 80%. Why? Because it's the same virus which is in weakened form or inactive form. I underline, it is weakened or inactive. Because most of the people are having the fear that we are going to take the same virus that will cause COVID-19. No, it is the weakened or disarmed or inactive form of SARS-CoV. There is nothing to feel afraid that you are going to take the same virus. It has been killed, it has been weakened, it has been disarmed, it is inactive. So there is nothing to feel afraid. Go and take the vaccine as long as this thing happens. And there are 2 doses of this vaccine also. 28 days gap is maintained for the 2 doses, and manufactured by Bharat Biotech of Hyderabad, and ICMR, a very big governing body is there, Indian Council of Medical Research. And this vaccine is using SARS-CoV-2 in a very weakened form, in inactive form. Also, one more thing, it is in refrigerated condition. The Sputnik V vaccine that has been there, it's freezed, means it is in powder form, but this one and Covishield is in liquid form. So this is, you can say, about the co-vaccine. Another important factor, the efficiency level or the efficacy level of this vaccine is a little bit higher than the Covishield, because here the same virus that will be used as a vaccine. Vaccination means it is when the idea is that your body is able to produce adaptive immunity, because the secondary immune response has a good immunological memory also. It remembers its enemy, that earlier I had been attacked by this virus and I have killed this virus. So the body's antibodies are ready with the army. So the second time when the real virus comes, it attacks. So this is not a vector-based vaccine also. Covishield is a vector-based vaccine. If you really want to know, go and watch my videos which are made on the vaccines, Sputnik V, and this one is about Covishield, co-vaccine. Now I am going to talk about the basic question that comes in our mind about the vaccines. Questions about vaccines. Can I take a vaccine of 2 different companies, means the 2 different doses? The first dose, say, of Covishield, and the second dose, that of co-vaccine. Now this question, when I'm answering, first of all I want to say it's basically specifically depending on the result of the research. Right now, theory, what does the theory say? Theory says yes, we can have it, and earlier we have seen success of such vaccines, the doses that are from different companies that has been used in case of hepatitis, in case of rubella virus also. So we can say theoretically it is there, but practically I will not be able to say at this stage, because at this stage we have to depend on the research and the results which are awaiting. So theory says yes, of course, but practically it's a question mark still now. Still now it's a question mark, but it can be answered with more research work on it. Next comes vaccines efficacy. Sputnik V claims that almost 91 to 92% efficacy rate. Covishield claims 70.6 to 70 to 80 range, and Covaxin claims 80% efficacy rate. With little bit of side effects will be there, but as far as Covishield is concerned, it is conditionally approved. In case of Covaxin, it is restricted use. Now this question that when a vaccine has to be taken, few factors are also there, few protocols are also there. That is in case of a pregnant woman, a lactating mother, or a comorbidity factor is also there. Comorbidity factor, if a person has multiple weakness, that is also a factor. So these are the factors, but otherwise, please go and take some vaccines. Do I am not talking about the social issues in this case. I am just talking about the science over here and the mechanism over here. So with this, I am going to finish up the video. If you have more questions on the vaccines, you can please write to me, and please do give me some likes and encourage. Hello viewers, I am back again with another topic based on coronavirus, novel coronavirus, nCoV which is subsequently known as COVID-19. You must be knowing in the recent time there are various strains or variants that are coming up of this virus. Now let me brief you one thing, that before I start this discussion on the new variants of coronavirus, let me take you a little bit back to that period of time when last year, 2020, initially when we came to know about the virus COVID-19, which is nCoV, we say, novel coronavirus, when it started all the way from Wuhan in China, and then slowly, gradually traveled to other countries, and then finally it reached India. In India, we came to know about it, then we started working on the vaccines. So there's a lot of stories about it. But right now, the story stands on this fact that we are coming to know about the new variants. So the topic of discussion will be on the new variants of COVID-19. There are several variants we have been known to. Earlier, we have been known to a very transmissible, very infectious variant of UK variant, then Brazilian variant, and then later on coming up more AP variant. Other variants are also there, and by the time you will be listening to this and watching this video, probably more new variants will be coming. So there is nothing new for a virus to mutate. Influenza virus, over a period of time, we have known about so many variants of influenza virus, and it has mutated. So mutation is means there is no law, no logic, nothing that we can ascertain the fact that no, this virus will not mutate. It's quite natural for a virus to mutate. The person's cells are the host cells. Host cells are being attacked by the virus, whose spike proteins containing, or you can say encoding, the pathogen, the antigen rather, hemagglutinin in case of viruses, in case of influenza virus it is hemagglutinin or neuraminidase. So it's going to encode the protein, and definitely it will be an entry route for the virus into the host cell or the host body. But what happens, our immune system is a very profound immune system. So when this person A is being attacked by this virus, the cell receptor, these are the cells, host cells. These host cells are producing antibodies that are denoted by YY, we say. It attaches to the spike proteins. Say this is the area of the spike protein where it is encoding the hemagglutinin is a glycoprotein. In this case, it is binding on to this protein, this glycoprotein, our antibodies. Let me draw it in this way so that you can understand. Host cell antibodies are binding on to the glycoproteins, that is the hemagglutinin, which is the antigen, and it is trying to attack it and kill the virus, so that the host cell is being safe. that we have known that to keep the host cell safe, this is the normal case. What happens? The antibodies that are there present in blood, our immune system is going to have those antibodies. These are neutralizing antibodies. Now the scenario is a bit changed over here. Now the virus looks for a survival route. It wants to attack the host. So it has to change itself a little bit so that our immune system is unable to recognize who is going to attack me. So our antibodies are good enough to identify the foreign invader. Antigens are actually small molecules, you can say, and these are carrying the hemagglutinin or neuraminidase, and these are going to serve as the infectious particle. So our antibodies are quite good enough to attack and cling to those antigens and then kill them. Even our vaccines have this responsibility when our immune system is trying to do it and unable to do so. Our vaccines are doing the same work. What our vaccines are doing? Our vaccines are also trying to attack those antigens by inducing the immune response which will be supplied by the antibodies. Vaccines will develop antibodies by first of all recognizing which antigen it had been attacked with. It has a good immunological memory also. It remembers its enemy, that yes, this was the virus, it had attacked earlier, and now when it attacks, I am going to bind it with the proteins and I am going to keep my host cell and my body safe. This is the work of the vaccine and our body's immune system. Virus is looking for an escape route because it knows that the host body, our body, immune system has a good immunological memory. So it will remember me. The virus is saying, yes, it will recognize me that I had attacked earlier and I was killed. So second time when I go with the same load, I will be attacked. So I have to dress myself in a very different way so that I can change myself. So what is that small change it does? Now the story of mutation begins over here. That yes, in RNA virus, RNA when we are talking about RNA, we mainly focus on these four letters A, U, G, C. In case of DNA, it is double-stranded, it is ATGC. In case of RNA, RNA exports, that is a single strand first of all, and it is all about AUGC. Now, what it does is, suppose it enters our host cell, means our body. And what is its work? A virus has the work to replicate, to reproduce, to make more copies. Now, what it does is, this is the RNA structure, means it's a very hypothetical thing. I have tried to represent it on the board, that yes, say its nucleotide sequences are somewhat like this. This is the original one. It tries to make multiple copies. Why? Because its motto is replication and causing damage to the host cell. Now what it does is, when RNA is trying to make its copies, at the time of replication, these RNAs, when it is trying to make multiple copies, there are some points where it is making some mistakes and certain changes are occurring, and those changes cause mutations. Mutations are small genetic changes that occur in the nucleotide sequences. Now what happens when it is a point mutation? Like in case of antigenic drift and all, we say it's a point mutation which is taking place in a single nucleotide of the nucleic acid. So in this case, let me not go to that part right now. Let me just show you what happens is, the host cell is being attacked by the virus. Now it has entered with its genetic material RNA and it is trying to make copies. When it is trying to make copies, that it makes some errors. Errors are the causes of mutations. Errors means some mistakes. Like earlier the sequence, and this is a very imaginative work I have done over here. So let's look at the sequence. As I have earlier stressed on the errors, errors, the small genetic changes, as it is RNA genome we are talking about. So naturally the changes will take place in RNA genetic sequence. It's not that the entire sequence gets changed. All genetic changes are not leading to these sort of mutations. There are slight changes that occur. Maybe a small change can even affect. What happens is, say the sequence earlier means when it was starting with this sequence AAG, UAGC. When we are talking about RNA, we cannot put the letter T because its RNA means AUGC. So I have just put in this sequence. Say this is the sequence, genetic sequence, and there are some, there are some mistakes made by this virus, and those mistakes, the small, are leading to small changes. Earlier it was AAG, UAGC, and now it has become, say at this point, there is a slight change. Instead of A, it's U. Again it is having A. Again it is having G. Then it is again, instead of U, it's having A. Then again it's having the same sequence. So while copying, this was the original one and this is the copied one. It's not that in all locations it has changed. It has changed only at some points. So small mutations which are occurring can lead to encoding proteins which are different type. That means the whole of the structure gets changed entirely. That is not the point. A small change can even be enough or sufficient to have the different set of spike protein. Earlier, the origination, when we started with the virus, it looked something like this. And you can see how it is looking. It's by the look only you can understand that yes, it was something looking like this. Now the coat proteins, means this change can lead to changes in the protein coat, and that will be instigating the spike protein. So in this case we found that small change at 2 points resulted a substrain of the virus. And what was the reason? The reason was mutation. Small genetic changes or errors that lead to different strain of virus and that causes mutation. Now in case of coronavirus, we are hearing double mutant, triple mutant. Mutation can take place at a certain point, or in case of novel coronavirus, the nucleotide is a very large one, so it's prone to make errors at multiple locations. It can make errors at different locations. So maybe genetic changes or mutations at one point several number of times, or maybe multiple locations errors it is making because it is lacking the proofreading activity that will be able to correct. Like when we make some mistakes, what do? Our computers, we make a spelling mistake. There is a spell checker option. It automatically checks. But in case of RNA viruses, that spell checker option like thing is present, that proofreading is present. But as the nucleotide sequence is a very large one, so it's prone, it's likely to make errors, and those errors will lead to mutations, and those mutations will be able to encode different set of proteins, not entirely, but some of the code proteins will be changing. Henceforth, we will have a new substrain of the same virus. As I have said, there are some other RNA viruses also. When I will be making a separate video on that, then I will be talking about it. There is hepatitis C, E, there is influenza virus, Ebola is there. So it falls under the peptide classification. You will find it under group 3, 4 and 5. You will find it. So I am not going into that. What I am focused right now is on the new variants, how the new variants have arisen. Now as I said, it's all due to the mutation, and mutations are small changes that take place in the genetic sequence, as I have shown you earlier. What the slightest change can alter, or it can encode a different code protein, that is the spike proteins. Henceforth, these spike proteins are when they have a different look, they look like a new strain or a new variant of virus. That is, we say double mutant, triple mutant. These are some of the words that I used. These are double mutants. It's originally from one we have come. We have got the SARS-CoV-2, and from there the substrains, you can say, have arising. So I'll just show you in a little bit in a different mode so that you understand this. Like say this is the genetic sequence. Earlier it was this. Say the pattern was like this. Now when a small genetic changes take place, say from here, this point, there is a certain change and we found it was over here, this change. This small thing is good enough and sufficient for that change that we found in the coat proteins, that is the surface receptors where you will find encoding the protein hemagglutinin or neuraminidase in case of influenza virus. That's going to code a different protein. So that's how we got the substrate. So from here, due to mutation, we may get a substrate like this. This is the original. SARS-CoV-2 has taken 1 year almost. If I am going to look at the duration, mutation is not a process which is going to be very rapid. It's going to be slow, gradual, but it takes place. And a virus is prone to mutations. It's nothing new for a virus to mutate. It will be giving more variants. And we need, then the question comes about vaccination, whether it will work or not. So if there is, in case of coronavirus, what happens is, there are genetic changes found in more than two sequence changes have taken place, more than that. So that's how we have got different strains.
Genetic changes, which we know of small changes, accumulations of these changes, accumulations of the mutations are resulting in antigenic drift. Antigenic drift is actually the causes mutation in a virus, which is enough, a small change is enough for the virus to code a different code protein. So you can say the spike proteins, and all the way what we have got due to mutations, 2 substrains of the same virus. The parent virus, which we started off, had the code protein which looks like this, and here you will find only at 2 points I have changed. It has got a different look. It's not the entire thing has changed. Maximum look is that of the parent strain, but 2 points you can say I have tried to give a different look. So a small change is good enough to create that the different code protein and get a different strain of virus. It's not a new virus, it's the same virus with a new strain or a new variant, and the cause is mutation or antigenic drift. Antigenic drift is means it can be treated with antibodies and drugs. It can be treated, and it takes some time to do it. In case of coronavirus, we have seen that over the year it has happened. And another substrate you can see, that is here I have tried to create for you so that you understand. Here it was at 2 points, here it is at 3 points, but still the look is almost similar. So it's coming from the same virus, new strains, new variants, and we have all the different strains. This UK strain, Brazilian strain, and AP strain, West Bengal strain. These are actually coming due to mutations. Mutations are causes for antigenic drift. Do not mix up with genetic drift at all. Genetic drift is a completely different phenomenon. What our immune system, when it is unable to deal with this matter, we have vaccines. Again, we need to see. Some of us believe that the new strain of virus will not be, means the vaccines that are coming out will not be effective. No, it's not that also. When we talk about a vaccine's effectivity, generally the approval of effectivity or the efficacy of vaccine comes only if it is above 50 to 60%. So our vaccines are far above that barrier. It's all about 90% efficacy. Some of the vaccines, like in Covishield, we know it's 72 to 70, yes, it's almost 72%. Then we have Biotech's Covaxin, which is also claiming 80%. Then Sputnik V says more than 90%. So our vaccines are good enough, even if the spike proteins get changed. Maximum spike proteins will remain that of the parent strain. Henceforth, our vaccines will be able to fight. Maybe the rate comes down from 90, the efficacy rate will come down to 75 or 70%, you can say. 72 can come down to 60 or 50. So it will be effective. It's not that the vaccines will not start working. It will work, but the efficacy rate will be lesser.
Now this antigenic drift, as I have explained, is not such a severe thing as the next one coming up. Now from the two substrains, we have got the parent virus which looks like this. From there we got two substrains of the same virus due to mutations, due to antigenic drift. I should write over here, this is antigenic drift. Mind it is not genetic drift at all. Genetic drift is a completely different phenomena. Now from here, now suppose these three separate strains can get an entry route into a person's body. Means a person is a host for the virus. Now the person A is having all the three substrains of the same virus. This was the parent strain, this is the A, this is the B substrain, and all the way it gets the entry route into the body of a person. So you can say in this case what will happen, it's not that the genetic material will be mixed up. No, there will be rearrangement of the genetic material of these substrains of viruses, and due to rearrangement or reassortment also you can say, you will get to see all the way a new virus coming up which is absolutely different from the parent strain. It is not similar to any other strain. It all has a different look, and this is known as antigenic shift. And when this takes place, it gives rise to another pandemic itself, and it's a severe one, fatal one. It's very rapid, and it's... See, for an antigenic shift, the necessity is what? The host, that is a person who is living, and immune system is unable to recognize those code proteins, those spike proteins, because those are the different strains that are coming up of the same virus, the original virus, giving rise to, as in my diagram I have tried to show you, from the parent strain we got two substrains due to antigenic drift, due to mutations. Remember, a virus needs a survival route, and for a virus to survive, it mutates. It tries to escape the immune system. Our immune system is very good. It has a good memory, immunological memory, to remember its foreign invader who had attacked earlier. So what it does is it tries to escape the immune system by giving a different look, and that different look is none other than a different strain. So that is the story of antigenic drift. But somehow these different strains of viruses, if it gets another person who is a healthy person and enters the body, the cell, as I said, that the cells are going to be reprogrammed. They are going to have the whole multitude of viruses, and when the cell bursts open, it infects other cells. And here in this case, it is not at all the genetic material will be fusing with each other. It's going to be a rearrangement that takes place of the three viruses genetic material, three substrates viruses of the genetic material gets rearranged, and a different form of virus takes place. The criteria for antigenic shift to occur is different substrates getting a healthy person's body. And in this case, will the vaccines work? Sorry to say, no, the vaccines, because why the vaccines may not work or will not work, the reason is it's a new form. The vaccines have been developed on the basis of the strains that have already been studied. We have studied the strains, and accordingly we have tried to develop the antibodies which can encode the spike proteins of that kind. Now if it is a different spike protein, whole arrangement is only changed, then how can those vaccines can start working? So this is a severe phenomenon. It's very rapid, it's very rare also, it's very rapid also. But when it takes place, it really becomes a pathetic state, very drastic state. So antigenic drift is due to that mutation, and antigenic shift is due to rearrangement or reassortment of the genetic material that takes place. And a new virus form, you can get it, of the genetic material. Okay, so I hope this antigenic drift and shift, there are more details on it which I am not able to cover in this video. I hope that you will be encouraging me with my work so that I can come up with all the videos which can be related to these topics and detailed account on antigenic drift and shift, where I will be talking about hemagglutinin and neuraminidase, how the activity takes place in a virus, and I can talk about it. For the time being, please support me, of course. Without your support, I am nothing over here, so it's your support which can help me bring you more of the material. Thank you for being with me.